Hey guys, welcome back to my channel. It's going to be a little bit of a different video today. We're going to be comparing mitosis versus meiosis because we know we all get a little bit confused sometimes. So let's have a look into mitosis first. Mitosis was first discovered by Walter Fleming in the year 1880. It is the cellular division of nucleated cells into two identical daughter cells. Hence, it only happens in eukaryotic cells. Mitosis has three main functions. The first function is repairing cells which have been damaged or undergone trauma. For example, if we fracture or break a bone, the bone must undergo mitosis to repair the damage and maintain its normal function. The second function is general growth and cellular reproduction by increasing the number of cells in a multicellular organism, such as blood and skin cells, which are continuously renewed to replace the ones that have reached the end of their lifespan. Mitosis is essential to ensure the daughter cells have the same exact function as their parent cell. The third function is for asexual reproduction, which can be used to produce exact copies of an organism. As observed in the regeneration of starfish, they have the ability to reproduce asexually by a process called fission, where a whole new starfish can be developed from an arm of a parent starfish. Next, let's have a look into meiosis. In 1876, Oster Hartwig discovered meiosis through his studies on sea urchin egg development. Meiosis is a process of cellular division involving the production of 4 unique daughter cells. Each consists of half the chromosome number of the parent cell. Its main function is sexual reproduction of organisms such as animals, plants, and fungi, produce gametes such as sperm in males and egg in females in preparation for fertilization of the zygote. So that's a bit of background information on mitosis and meiosis. We're now going to explain the 5 phases of cell division. I stands for interphase, involving DNA replication. P stands for prophase. This is where the duplicated DNA is paired. M stands for metaphase. The chromosomes now meet at the metaphase plate. A stands for anaphase. Now the chromosomes separate. Finally, T stands for telophase, where the 2 daughter cells split into 2. It's important to note that there are no real distinct phases, however, only prophase to telophase is included in mitosis. Interphase is the phase of the cell cycle which a cell spends most of its life. During this phase, uncondensed DNA strands known as chromatin starts to replicate within the nucleus in preparation for mitosis.
20 hours later.
In prophase, chromatin condenses into chromosomes which consist of 2 sister chromatids joined by the centromere. Microtubules start to form spindle fibers and the nuclear envelope breaks down as the cell prepares to enter metaphase. In metaphase, spindle fibers are fully formed with microtubules attached to each sister chromatid. This allows the double-stranded chromosomes to align on the equator of the cell across the metaphase plate. Anaphase involves the separation of sister chromatids as the centromeres holding each chromosome pairs divide. New chromosomes, each derived from one chromatid, move towards the spindle poles. Telophase begins with the presence of the plane of constriction. Nuclear envelopes reform around the 2 groups of chromosomes that have reached the spindle poles on both ends. The spindle fibers then start to disappear and the chromosomes decondense into chromatin. However, mitosis will not be completed until cytokinesis takes place. The mitotic cell divisions conclude with cytokinesis, a process of dividing cytoplasm, organelles, and plasma membrane, which results in 2 identical daughter cells. On the other hand, meiosis is split into 2 separate stages, so let's have a look into meiosis 1 first. Interphase 1. This phase is the same as it is in mitosis. The uncondensed chromatin begins to replicate in the nucleus. Prophase 1. As the nuclear envelope disintegrates and spindle fibers start to form, the homologous chromosome lines up next to each other, forming a bivalent structure. The DNA exchange then occurs between the homologous chromatids, rendering homologous chromosomes which are no longer identical. Metaphase 1. The homologous chromosomes line up at the equator of the cell rather than sister chromatids as shown previously in mitosis. This orientation is completely random and is independently assorted, creating a larger genetic variation. Anaphase 1. The chromosomes move apart from one another along the spindle fibers attached to the centromere. At this point, the chromosome is still double-stranded. The separation of homologous chromosome results in having only one member of the homologous pair in each cell. The first phase of meiotic division concludes as the cell moves into telophase 1, where the cleavage furrow and nuclear membrane forms. followed by cytokinesis 1, the cell membrane formation of two daughter cells, each containing haploid chromosomes, which then decondense into chromatin again. After a period of interkinesis, the two daughter cells simultaneously start the second meiotic cell division as they enter interphase 2 without undergoing DNA replication. Prophase 2. The chromosomes condense again as the nuclear envelope breaks down. The spindle fibers form at the poles of both daughter cells and attaches to the centromeres of the sister chromatids. Metaphase 2. Here the chromosomes line up singularly at the equator of the cell. Anaphase 2. The sister chromatids separate as they move along the spindle fibers to reach the spindle poles. Telophase 2 takes place as the nuclear membrane reforms around the chromosome and the spindle fibers disappear. Cytokinesis completes the second phase of the meiotic division. We now have 4 genetically different haploid cells. There are some unique features that occur either only in mitosis or meiosis. Let's zoom in on the features in mitosis first. Most somatic cells in our body take the mitotic cell division route, such as skeletal muscles and skin cells. The cell undergoes one set of cell division stages where the sister chromatids separate during anaphase and 2 identical daughter cells are produced from that one parent cell. Moving on to meiosis. It is a process of germ cell division for gamete production. It has 2 stages of cell division occurring one after the other. Unlike mitosis, the completion of this process results in 4 unique daughter cells that only have half the number of chromosomes from their parent cell, known as haploids. Thereafter, the homologous chromosome pairs line up and independently separate at the metaphase plate. Instead of sister chromatids separating in anaphase seen in mitosis, the homologous chromosome synapses and recombination of DNA segments between non-sister chromatids occur in meiosis. Despite having different features, both mitosis and meiosis are processes of cell reproduction. Both involve 5 basic phases of cell division, namely IP-MAT, that occurs once during mitosis and twice during meiosis. The duplication of DNA for mitotic or meiotic cell division takes place during similar phases, known as interphase for mitosis and interphase 1 for meiosis. In metaphase of mitosis and meiosis 2, individual chromosomes align at the metaphase plate in preparation for sister chromatids to separate. So that's it for our lesson on mitosis and meiosis. I hope it was helpful and that you enjoyed watching. Don't forget to give it a thumbs up and hit that subscribe button. See you all in my next video. Bye!